Hey folks, today we're going to be discussing and dissecting the Atlantic sharp-nosed shark, by far the most common coastal shark species we have here in the northern Gulf of Mexico. And if you've ever caught a shark in your life, there's a good chance it may have been one of these. So Atlantic sharp-nosed sharks are pretty easy to identify. You can see they have these white splotches kind of haphazardly along the sides of their bodies, all the way up here, even up to underneath their snout a little bit. They have this dark grayish color on the dorsal surface, which blends into a white color on the ventral surface. So again, this is a very common coastal species. It's also a member of the family Carcharodontidae and the order Carcharodontiformes. And what that means is that this is a ground shark or a requiem shark. So that's a very large group of sharks that all have this more or less kind of sharky look to them, and they share lots and lots of features in common. Before we get into further discussion about the details of this Atlantic sharp-nosed shark, let's talk about some of the features of this fish that are common to most sharks we have here in the Atlantic Ocean. So starting at this end, this is the anterior end of the fish, and conversely, this is the posterior end. This side in general is the dorsal side of the fish, characterized by the primary dorsal fin and the secondary dorsal fin. This surface of the fish is the ventral surface. So again, this dorsal surface has both a primary and a secondary dorsal fin, and we call these median fins because they're located along the medial line of the fish. On the other hand, this shark also has two sets of paired fins. These are the pectoral fins, located in the pectoral region of the fish, just like you might imagine. These are the pelvic fins, paired fins, located on the ventral surface, located in the pelvic region of the fish. Last but not least, this is the anal fin. Not all sharks have anal fins, but members of the family Carcharodontidae, or the order Carcharodontiformes, have an anal fin, and that is a median fin. It's not a paired fin. So median fin, paired fins, median fin. They also have what are called pre-anal ridges, which are these two little tough ridges of skin right down here. This is the caudal fin, and you'll notice it has two distinct lobes. This is the upper lobe of the caudal fin. This is the lower lobe. This is a notch characteristic to many sharks in this family, the family Carcharodontidae. It's called the precaudal notch. But you'll notice this is what we refer to as a heterocercal caudal fin. In other words, the upper lobe of the caudal fin is significantly longer than the lower lobe. So starting here and moving towards the posterior end, we can see several things right off the bat. This shark has paired nares. So here's one nostril, here's another nostril. These have both in-current and ex-current openings. So water comes in one portion of the nostril, goes across a series of sensory epithelia, and comes out the other opening here. Another feature you'll notice on many sharks, most sharks in fact, are these little tiny pores called ampullae of Lorenzini. You can see they're very extensive all over the snout of this fish. Some holes much smaller than others, all around here, around the eye, even here around the corners of their mouth. You can count numerous little pores that make up the electroreceptive system for this shark. And again, these are called the ampullae of Lorenzini. And the shark uses this sense to detect weak electrical stimulus that are in the ocean, emitted by animals, struggling animals, and things of that nature. You also see, like vertebrates that we're familiar with, this shark has a very typical eye for a vertebrate. And you'll notice, like other members of the family Carcharodontidae and the order Carcharodontiformes, this fish has a nictitating membrane. So a thin protective eyelid, if you will, that comes up over the eyeball and protects the eye when this fish is feeding or when it's mating or things of that nature. And if you ever have the opportunity to get up close and personal with an Atlantic sharp-nosed shark, you'll see they have relatively well-developed labial furrows, which are these little lines right around the edge of the mouth. So we talked about the nose. We know the two nares for this shark are right here. We talked about its eyes. It's got a pair of eyes right here. We don't tend to think of sharks as having ears, but in the strictest sense they do. We refer to them as endolymphatic pores, which on this guy you can barely see, and they're located on the top of the head, right there and right there. And these allow the shark not to hear in the same sense that we hear things. They use these more for a sense of balance and maintaining homeostasis. Now, sharks don't have a special bony plate that covers their gills. They instead have these gill slits. And in this particular shark, we can see 1, 2, 3, 4, 5 gill slits. 5 gill slits are characteristic of the vast majority of sharks. Some sharks, called the 6-gill sharks, have 6 gill slits, and then there are some that have 7 gill slits, and there are even some that have 4 gill slits. But the vast majority of the sharks have this condition right here, 5 gill slits, and these simply cover the delicate gills that are underneath this portion of the skin. Of course, we know sharks don't have bones, but they do have cartilaginous supports that support these appendages. For example, here we have the left pectoral fin, and if you can look at this underneath the light, I'm not sure if you can see that or not, but you can see these striations right here. These are ceratotrichia, the thin cartilaginous supports that provide structure for this appendage. Now, another special sense that this shark has on the outside of its body, located along the skin, is a sense that it uses for mechanoreception. And you can get a good look at that line, called the lateral line, right along here. And so let's imagine for a moment that you're a fish swimming through the water, and your fins are moving back and forth, and those fins are moving water away from the fish, and that water is propagating through the water column. It's moving across the water column and creating a pressure wave. And that pressure wave, or that mechanoreceptive stimulus, is something that this shark can detect using this lateral line system that we have right here. So we've talked about the eyes, the nose, the endolymphatic ducts, the fins, the ampullae of Lorenzini that we see around here, the lateral line that we see in this general region. But the question remains, how do we know if the shark is a male or a female? Now, unlike a red snapper, which you have to open up in order to tell if it's a male or a female, sharks give you a way to tell externally. So these are the pelvic fins of this animal right here, and we know that this is a female because she does not have claspers. Claspers would be 2 long appendages coming out of this region of the fish. So we'll dissect a male in a bit, but we can definitely tell by looking at this fish that she's a female. All right, so we've talked a little bit about the external features of this Atlantic sharp-nosed shark, but our main objective today is to open it up and to talk a little bit about the internal anatomy, so a true dissection. Now, like any dissection, there are many ways we could open up this fish. We can make a cut along the ventral line here, the midline of the ventral surface. My preference is to gently pull up on this left pelvic fin, make an incision here, and essentially follow this gradient where it changes from white to gray all the way up to the left pectoral fin and then down to here. Now, this pectoral fin is marked by what's called a pectoral girdle, or a piece of strong cartilage that supports these 2 pectoral fins. So this general area is the gastrointestinal cavity, and that's really what we're looking to open up. So by making this cut like this, around this fin and down, this section will lay open, and then we can discuss some of the internal anatomy. So my philosophy when I'm dissecting is to make shallow cuts, right? So just like going to the barber and getting a haircut, They can always cut off more, but they can't glue your hair back on once it's been cut off. So we're going to make shallow cuts. Our intention is to not puncture into the internal anatomy in here, because that's exactly what we want to get a look at. So shallow cut here, smooth cut along this general area up till there. And again, I can always go back and make that cut a little bit deeper. The skin is going to be the toughest part of this fish, of course. Now I'm going to lift up on this girl right here. Especially here, I want to be shallow because it's easy to cut a little bit deeper than you intend, and that really makes a mess. Now, when we first started discussing this female Atlantic sharp nose shark, I suspected that she might be gravid. And what we mean when we say gravid is just carrying embryos of some stage. And as you can see right here, that's exactly the case. So we're going to discuss that towards the end. We're going to start with some of the organs that you would first see when you open up any shark. And the most dominant organ you're going to see in a shark is right here. And this is the liver. And there's lots of really interesting things about this liver. If you look at this fish and look at the size of this liver, that's an enormous percentage of the entire mass of this fish. And what's interesting is this is only part of the liver. So there's one lobe. And there's another lobe. So suffice it to say, sharks have a relatively large liver compared to the size of the overall animal. And we'll talk later about why that is, but for now, let's just suffice it to say that has to do with buoyancy. Now, one of the traits that make sharks different than other bony fish is their lack of a swim bladder. But since they don't have a swim bladder, they have to compensate for that lack of a buoyancy control mechanism in another way. And they do that in part by having a very large liver filled with lipids. Lipids are very dense. They help keep the shark afloat in the water column. So a shark's gallbladder is located on the ventral surface of the liver, and there's a common bile duct that goes from that gallbladder down into the duodenum. So that's the way that the shark is able to get bile from the gallbladder down into the digestive system. And you can see here some of that architecture. So again, two primary lobes of the liver. This is actually the third lobe of the liver, but it's insignificant, as you can see. They take up a massive amount of the space of the internal cavity of this shark. And I want you to remember that as we go through the remainder of the internal anatomy of this fish. So this is a given size, right? There's only so much room inside the gastrointestinal cavity of this shark. This liver is hogging all the space inside this animal. It's taking up way more space than it has any right to. Consequently, all of the other organs inside this shark, inside the gastroperitoneal cavity of this shark, have to accommodate. They have to have special adaptations to account for the fact that the liver is taking up so much space. Since it's hard to see what else is going on outside of this, let's go ahead and discuss this. Unlike mammals, certainly unlike humans, sharks have more than one uterus. In fact, they have two uteri. So here we see the left uterus, and down here we can see the right uterus. Both of those uteri join down here in the cloaca. And this is a really, really interesting thing because this shark at the same time is developing these embryos in utero. So a handful of developing sharks in the left uterus, a handful in the right uterus, and at the same time, its ovary is filled with what are called vitellogenic ova. So these are ova that are filled with vitellogen and are being prepared to be the next cohort of little sharks that develop within each uterus. All right, so now we're going to remove this liver. There's a single ligament up here where it's attached. And again, just take a moment to appreciate how absolutely massive this liver is with respect to the rest of the body cavity for this shark. So moving through the rest of the internal anatomy of this fish, of course, here's the mouth. Here we see the esophagus. And you can see how that tissue is very different in color and texture from this tissue. So this is the upper part of the stomach, the cardiac stomach, and this is the esophagus. So what we're going to do now is make a cut right here at the esophagus. I want you to take a look inside there and see how thickly walled that esophagus is right there. And if you think about it, this is a very valuable adaptation for a shark as it's struggling to get prey into its stomach, things that have lots of sharp scales and spines and fin rays. This helps protect the rest of the internal anatomy from those sharp projections. So here we see the remainder of the stomach, and here we have a dark organ. Does anybody want to take a guess as to what that is? Concentrate on this dark red organ, and that's the spleen. So this is really incredible if you think about how big this liver is compared to the size of the gastroperitoneal cavity. It's immense. And as a consequence, all of the other organs inside this shark have to compensate accordingly. They have to come up with unique adaptations to reduce their size, given that the liver is taking up all the space. So it begs the question, why is the liver so big? And of course, we know that sharks don't have a swim bladder. We know that their bony fish relatives, red drum, speckled trout, red snapper, they have a gas-filled swim bladder that acts as a buoyancy control device. And they can regulate the amount of gas in that swim bladder to change their position in the water column, either up or down. Now, sharks don't have swim bladders. In fact, we know that's one of the things that separates them from their bony fish relatives. So, one of the reasons they have such a large liver is to aid in buoyancy compensation. Specifically, this relatively large liver is filled with very low density lipids. Let's do a little experiment. Let's see what happens when we put this liver in water. Well, sure enough, it floats. So having such a large liver that's filled with such low density lipids gives this shark a little bit of extra buoyancy, which is exactly what you would expect it would need given the fact that it doesn't have a swim bladder. We know this is the mouth right here. We've discussed how thickly walled this esophagus is and why that might be useful. This is the stomach, the entirety of the stomach of this fish right here, and this big red organ is the spleen. This organ right here is the pancreas. Notice the color, it's sort of a tan, pinkish color. On a fresh specimen like this, the pancreas is somewhat mottled, and it's usually in this sort of a shape, sort of a V shape like this. So all of this is the pancreas. Now, you've noticed these tissues here, this very clear suspensory tissue that's holding all of these organs in place. So all of this suspensory material is called mesentery, and its function is to simply hold these organs in place. So here again, we have the pancreas and the spleen. So if this is the stomach empties into this organ right here, which for this particular shark is called a scroll valve. You may have heard of an analogous organ called a spiral valve, which we see in sharks like white sharks and spiny dogfish. In carchariniform sharks, or ground sharks, or the requiem sharks, we expect them to have a scroll valve. So esophagus, stomach, scroll valve, all the way down here to the rectum. So I'm going to make a single cut right there, and then another cut up here. And let's lay this whole system out. Now that we know what these organs are, let's take a closer look and see what some of their interesting adaptations are. So we'll start by making a longitudinal incision down the stomach. And what we see here is really, really interesting. Lots of very convoluted tissue. And these are called the rugae. These are the rugae of the stomach. And as you can imagine, their function is to increase the surface area available for digestion. So if your entire stomach has to fit in this small little package, well, perhaps that's not enough area needed to digest your food adequately. So what this organ has done in adaptation is created these structures called rugae. So without increasing volume, the volume of the stomach is set. This is a way by which the shark can increase the surface area available for digestion, which is a really neat feature. Now we're going to go all the way to the terminal end of the stomach here. And we can see that the stomach in this particular shark is empty, which is not terribly unusual. We expect about 50% of the sharks that we examine to have empty stomachs. And that's even higher when we're talking about pregnant females like this one here. That's a neat adaptation for the stomach. Now we're going to take just a second and look at a similar adaptation in the scroll valve. So again, the volume of the scroll valve is very set and very minimal. However, by having this scrolled architecture, it again increases the surface area available to this shark for reabsorption of water and minerals. So a really neat adaptation. So at this point, we've opened up our shark. We've seen that massive liver that's taking up so much space. We've removed that liver. Then we talked about the digestive system, starting with the esophagus, going through the stomach, and into the scroll valve. That stomach and scroll valve and esophagus are all held in place with mesentery. And within that mesentery, we talked briefly about the spleen and about the pancreas. So now that all of that material is removed, we can take a better look at the reproductive system for this fish. So again, this is the anterior end. This is the posterior end. This is the left uterus. This is the right uterus. And here's the ovary. And again, these are vitellogenic ova. In other words, they are eggs that this female has selected to further gestate. So once these pups undergo parturition, which means once they're born, these eggs will make their way into the uterus and start to develop the next brood. So let's take a moment and remove each uterus in turn so we can get a closer look. Now, this uterus is held in place by mesentery, but since it's uterine mesentery, it has a slightly different name, and it's called mesovarium. So I'm going to gently pull along the mesovarium here. And again, that's the left uterus. And you see an interesting organ right here. That's called the oviducal gland. So we can see here the oviduct and the oviducal gland. The oviducal gland is where the sperm and the ova meet and fertilization takes place prior to the egg making its way into one uterus. Now let's take out the right uterus, again pulling along the mesovarium. And as you might have predicted, the right uterus also has an oviducal gland. Okay, so now that we have both of the uteri out, let's take a look at what these little pups look like on the inside. So again, a very shallow cut, lifting as I cut. And right away we can see some really interesting features. So what you see right here is the umbilical attachment. Again, this is an Atlantic sharp-nosed shark, and Atlantic sharp-nosed sharks are placentally viviparous. And what that means is that they give live birth, but that live birth is facilitated by an umbilical connection. So this developing embryo is getting nutrients directly from its mother through this umbilical cord right here. So let's get a better look at exactly what that looks like. Here we can see what is essentially the belly button of this small shark right here. And the remnants of the egg, where the egg has made an attachment to the placental wall inside this uterus. So again, just like a mammal, this mature female shark is nourishing her developing embryos through a direct connection via this placental attachment and this umbilical cord, which is really pretty fascinating. And each of these sharks has its own egg envelope inside this uterus. You can see this thin membrane over this individual here. We remove that. And then one more. And again, each of them is encased in their own special membrane. Notice those little bitty villi, those little bitty hairs along the umbilical cord. So after removing those pups from the 2 uteri, we can see a total of 6 individuals. And just coincidentally, 3 of them are male and 3 of them are female. So again, if you think about the size of the liver of this fish, the size and the biomass of all 6 of these pups, all jammed into this really, really small space here, that's certainly a lot of load for this female to be carrying around. A couple more organs we want to talk about briefly now that we've taken out the liver, the gastrointestinal system, and the majority of the reproductive system. Keep in mind, this is the ovary here with these big vitellogenic eggs. What we haven't mentioned are 2 interesting organs right here called epigonal organs. We believe this is part of the circulatory system and aids in the production of red blood cells. And last but not least, we see a little purple organ right down here. And that's called the rectal gland. That serves to eliminate excess salts and plays a role in osmoregulation. Now, if you think about the organs inside of a typical vertebrate, you'd realize that we haven't yet discussed the kidneys. On most fish, but certainly in these sharp-nosed sharks, the kidneys are located right here along the ventral surface of the vertebral column. So I'm just going to push down right in this area. Here you can see the kidney for this animal. Okay, so we know that sharks are chordates. In fact, they're vertebrates, and so we expect them to have a backbone, which is a bit of a misnomer because we know that sharks don't have true bones. They're made entirely of cartilage. So I'm going to show you what the vertebral column of this animal looks like. So here we see the vertebral column. I've made a cut on either side, and now I'm going to remove a portion, show you what it looks like. I'm cutting between individual vertebral centra, right here, and then again, right along there. And once more, right along there. This is the notochord, essentially the spinal cord for this animal. And these are the individual vertebra, the individual vertebral centra. They look just like the ones in your own back. So now we want to take a look at some of the musculature, and a really good place to do that is the caudal region of this fish. So I'll show you exactly what I'm talking about. We're going to make a cut here. And what we see here are these tight bundles of muscle. Each individual muscle fiber is called a myomere, and they're connected with tissues called myosepta. And what I really wanted to show you here is some of the architecture. So we can see white muscle here. Notice that most of this muscle here in this fish is white. But on the outside we see a small band of red muscle. Right here and here. So here we can see that notochord. And here we see the vertebra. So we talked about the gastroperitoneal portion of the shark. Now we're going to talk about the pericardial portion of the shark. And they're separated by a strong cartilaginous structure right here called the pectoral girdle. So what I'm going to do now is cut through that. You can see it's a little bit of resistance right there. You can see that hard cartilage. And this is the region where the heart is. Again, remember, these are the 5 external gill slits, which provide an opening from the outside to the gills, which we can see right here. So we're going to make a few cuts, open this up a little bit more so we can get a better look. Here you can see what these gills look like, the gill arches, the gill filaments. So here we are on the dorsal side of the shark on the anterior end, and what we're going to do is remove a bit of tissue and the top little bit of the chondrocranium, a membrane there, so we can get a look at where the brain sits on this fish in relation to its eyes, which are right here, and its nares, which are under here. So let's do that now. And here we can see just the very, very top part of the brain. Let me cut a little bit deeper. There we go. Now I want to give everyone an idea of how the jaw sits in relation to the rest of the cranium, which is on the dorsal side. So we're on the ventral side of the fish right now, and we're just going to get a closer look at the jaw. So here you can get a really good idea of this bottom part of the jaw here. And we're just going to work our way around the mouth. And I want to point out 2 very interesting aspects right here. And right here are the orbital processes. This is one of the only real tangible connections that the jaw has to the chondrocranium for this fish. And this is one of the ways, this is one of the mechanisms that allows this shark to have a very protrusible jaw. So let's just keep on cutting around. So let's fast forward now and take a look at what a cleaned version of this looks like intact. So attached to the entire vertebral column here, caudal fin here, so this is the posterior end, dorsal surface of the fish, Here's the anterior end and the dorsal surface up here. So what we see very clearly here is the jaw and how the jaw attaches to very few points, really. Here we see the palatal quadrate, which is the upper portion of the jaw, and then the Meckel's cartilage, which is the bottom portion of the jaw. Here we can see capsules, little areas here where the nares, you would expect to see the nares in a live shark. Here, here, and then the eyes would sit right about here. So again, we can see exactly how that jaw attaches to the bottom portion, the ventral surface of the condocranium. And here we get a really good look at where we saw the brain sitting when we did that, we removed that piece of skin. Notice here some cleaned versions of vertebra, and they look exactly like you would expect a typical vertebrate vertebral centra. So here they are individually, here and here. And then of course here you see them connected along the entire body of the fish. All right, this is great. So earlier we looked at an Atlantic sharp-nosed shark, a female Atlantic sharp-nosed shark, one of the most common fish in the Gulf of Mexico, certainly the most common coastal shark in the Gulf of Mexico. And now we want to pivot and look at another very common small coastal shark that we have in the northern Gulf of Mexico, and this is called the Blacknose shark. So first you'll notice there are many similarities between this and the sharp-nosed shark we saw. They both have roughly the same kind of body shape. They both have more or less the same placement of their fins, same overall coloration. But really, when you start looking at them closely, you'll notice several very important details that distinguish these two species. So first of all, taking a look on the sides of this shark here, left and the right sides, you don't see any white speckling at all, so no little white dots. You'll notice here some really neat coloration right in through here and really up in this region right here. Now, if this shark were alive, this would be a much more pronounced kind of yellow copper tone, and that's really one of the distinguishing characteristics of the Blacknose shark. But as the name suggests, it does have this little dark black smudge right on the edge of its nose. Notice the Blacknose shark does not have those well-pronounced labial furrows that we saw with the sharp-nosed shark. So, no noticeable labial furrows, presence of a black smudge, and different tones, especially on this portion of the fish. But the main reason we want to talk about this Blacknose shark is to give you an idea of what claspers look like. So shown here are the claspers, the two intermittent organs present on all male sharks. We're going to go ahead and open up this guy and run through that internal anatomy again. So if you remember when we looked at the Atlantic sharp-nosed shark, at this point you could already see the left uterus and the 3 developing embryos that were already inside here. So this is what a much more typical shark looks like. You open it up and the first thing you notice is this large left lobe of the liver. We're going to peel back here. You can see right here is a great example of the gallbladder. In this case, the gallbladder is filled, and it's filled with a substance called bile. That bile will travel through a bile duct from the gallbladder into the duodenum, or for our intents and purposes, into the stomach. So we're going to gently remove one side of that liver, come around to this region, take out the other side, twist right here, and get that out of the way. we can see how much room that liver is taking up in the body cavity of this fish. And just as a reminder, these sharks do not have swim bladders. In other words, they don't have an internal way to control their buoyancy, unlike something like a red snapper or a red drum. So they have to come up with other strategies to help themselves stay neutrally or positively buoyant in the water column. One of the ways they do that is by continually swimming, and that works for many species. Another way in which they do this is by having a very large liver full of squalene, which is a very low density lipid. We've removed the liver. Now we come back to the stomach. You can see here the esophagus. I'm going to make a section right here and remove that. You can see this tissue is much more frozen than it was for the sharp-nosed shark, but this again is mesentery, a clear tissue that's holding these organs in place. Here we see this deeply dark purple organ, which is the spleen. And on this side, you see the pancreas. And it's worth noting that the internal architecture of these two fish is very similar. And that makes sense because they're in the same family and thus in the same order. Now, if we were to look at a different species like a mako shark or a thresher shark that's in a completely different order, we would expect to see more stark differences. But take a look here. Here's a really good example of how thickly walled that esophagus is. So just like that, we removed the digestive tract and several of the organs that were embedded in the mesentery. And that gets us to what I wanted to focus on with this black-nosed shark, which is shown right here. So if you'll remember, when we got to the Atlantic sharp-nosed shark at this stage, what we saw was a big uterus over here and another big uterus over here filled with pups. Of course, that was a female. Now we're looking at a male. So what we see here is the left and the right testis, and I'm going to remove both of those and their associated epigonal organs. So now I'm pulling along this mesentery, which in this male, at this part of the body, the mesentery that holds the testis in place is called the mesorchium. Coming all the way down here, and before I pull it out, I want to show this one organ here, just as a reminder. This is the rectal gland. This is the rectal gland. This is the remainder of the epigonal organ. And there's some epigonal organ still on the ventral surface of these two testes. So now that we've removed the testes, we can see here very clearly the seminal vesicles, which is where sperm mixes with seawater prior to being flushed through this opening here into a clasper, which then gets inserted into the female cloaca. All right, so we've had a chance to dissect some sharks. Now we want to switch gears for a minute and look at some skates and some rays. So just like the Atlantic sharp-nosed shark and the black-nosed shark that we talked about earlier, they have a lot of similarities, and to tell them apart, you really need to look at the details. Well, shown here are 3 elasmobranchs, and remember we've decided that elasmobranchs are sharks, skates, and rays. So shown here are 3 elasmobranchs that on the surface look somewhat similar, but when you look at the details, they're really quite different. So let's start over here. We have a young cow-nosed ray. Notice the very long, slender, whip-like tail, and you'll notice that the sting, which is what you call the stingray barb, it's technically referred to as a sting, is located at the base of the tail right here. Let's flip it over. Here's the ventral surface of the ray. This is one thing that separates rays and their closely related cousins, the skates, from sharks. Rays have gill slits on the bottom. Skates have gill slits on the bottom. Sharks have gill slits on the dorsal surface. So, cow-nosed ray. Here we have an Atlantic stingray. This is perhaps one of the most common stingrays in the western North Atlantic Ocean. You can see here this really pretty shape. You can see the ceratotrichia coming out this way. And keep in mind that the wings of both this Atlantic stingray and this cownose ray are large modified pectoral fins. So like another elasmobranch, like a shark has the pectoral fins that we examined earlier, these stingrays also have pectoral fins. They're just fused more generally to the body, and they're enlarged and modified. Here we see a relatively pointed snout. Flip this guy over, and again we see gill slits here and here. So on the surface, this animal looks very similar to this animal. They both have a pointed rostrum. You can see clear rostral cartilage here. Something somewhat similar here, but truth of the matter is this is an Atlantic stingray and this is a roundel skate. So here we have ray, and skate. So you might ask yourself, well, what's the real difference between a skate and a ray? And there are a few things. First of all, rays are the only ones that are going to have these. These are stings or stingray barbs. Skates do not have stings. Another major difference between rays and skates are that rays all give live birth. Skates, on the other hand, all produce these. Now we know these as mermaid's purses. This is the egg case of a skate. Now lots of skates have egg cases that look somewhat similar to this, with these kind of interesting tendrils that help the egg case secure itself to substrate. But there are differences amongst the skates in the types of egg cases that they make. Finally, skates have a much thicker, well-developed tail, and what we can see here are relatively well-developed first and secondary dorsal fins, whereas these stingrays have very thin, whip-like tails. So that's another general characteristic that separates at least these stingrays from skates as a whole. So now that we have a better understanding of the difference between rays and skates, let's go ahead and open up a ray and see what their internal anatomy looks like. So here we're looking at the ventral surface of a blunt-nosed stingray. You can see here 1, 2, 3, 4, 5 gill slits on the left, 5 gill slits on the right. Furthermore, you can tell that this is a male. This is a large male. Blunt-nosed stingrays don't get a whole lot bigger than this, so this is a pretty big specimen right here. Now what we're interested in is opening up that gastroperitoneal cavity, which is essentially going to be right in here. So here we have it. Again, this is an elasmobranch, so it does not have a swim bladder. So as we expect, now that we've looked at the Atlantic sharp-nosed shark and the black-nosed shark, we expect the liver in this animal to have a fair portion of this body cavity sewn up. So we remove one lobe and then another lobe, and indeed we can see that that is absolutely the case. The liver is taking up a lot of room in here. So one of the interesting things that I wanted to highlight that is different about this ray than the two sharks that we looked at is right here. Down from the mouth, esophagus, here we have the spleen, relatively small stomach, but then we can see here that this intestinal tract is very much different than the scroll valve that we saw in the carcharodonid shark. So let's take a look at what this looks like on the inside. And what we see here is very interesting. This is a spiral valve. So if you remember with the carcharodonid sharks that we looked at earlier, the idea was to expand the amount of space available for reabsorption of water and nutrients. And in this case, we're talking about that same exact strategy, but with a different mechanism. So rather than having an intestinal tract that rolls out like this as a scroll, This is spirals. So it's just like a spiral staircase. You can trace the path of fluid as it goes in and around and around and around. So again, another pretty ingenious mechanism for increasing the amount of surface area for digestion and reabsorption when in fact the liver is taking up a lot of space in this animal.